today's hyperlipidemia.
Yep.
Let's talk about it.
Yep. Today's hyperlipidemia, dyslipidemia is.
Okay.
Because you can have high and low lipids. I call it hyperlipidemia because really the stuff I'm talking about is used for reasons.
Oh. Good. Take a massive
All right, guys. So it looks like it is 1:30, so we are going to get started because we have a lot to get through today. So a couple of housekeeping items. If you haven't checked your email at all, the exam 1 grades are posted. Okay. The average was an 85%, which was really outstanding. So kudos to you. As a class as a whole, I think you all did incredibly well considering the new circumstances that you had to take the exam under, right? And for some, it's easier to get used to than others. Okay, you will get used to it. I think that, we're moving in the right direction. So very happy about that. If you did not do as well as you would have liked to, please don't hesitate to schedule an appointment with me. We can talk, not only content, but also learning approaches outside of class. That's really important. Or if you want to do an exam wrapper and review your exam, we can kind of take a look at the types of questions that you missed and how we might use that information to better shape your study for the next exam, right? Because there are definitely things that you can do to improve that, okay? Tomorrow, it looks like Dr. Naeem has got her recorded lecture up, okay? One of the things I will say that I forgot is that typically I don't finish this material in one day, okay? I didn't mention that, but you don't have to come in. So I think we'll see how far I get, okay? And what I might have to do is do like a Zoom session tomorrow afternoon for 20 minutes or whatever. If and again, I'm not taking attendance, so you can come or not, and I'll record that as well too, as long as I can remember to do that. And but we'll see. I might be able to make it through most of this stuff. We'll see what kind of questions there are and where people are at. Okay. So again, if we don't, I'll let you know at the end of class what we're going to be doing. Other than that, I think we should be in good shape. Okay. Any questions? All right, so we are now moving on to our second disease state in the cardiovascular block. And I mentioned to some of you probably on day one that the way that the diseases are sequenced here, okay, is very purposeful. Because these first two diseases, hypertension and dyslipidemia, okay, for those of you that are familiar with that term, again, the lay term is high cholesterol, right? These are kind of, I think of them as baseline disease states that people can get, right? And which, if you do have them and they're not managed appropriately, can lead to some of the more severe cardiovascular diseases that we're going to talk about coming up. Things like coronary artery disease, angina, right? Heart attack, stroke, those sorts of things. Ultimately, heart failure or arrhythmias. So again, the sequela or series of events that, the series of diseases we're talking about are really listed in a particular order for a particular reason. So today we're going to talk about, again, the other kind of baseline disorder here that, again, can ultimately lead to other problems if not managed, and that is dyslipidemia, right? Okay. I have it referred to as hyperlipidemia because I'm really talking about drugs that treat high lipid levels, okay? But the term dyslipidemia just means the body is unable to regulate lipid levels in a normal way. And they can be, in some cases, most of the time they're abnormally high, but for some patients they can be low. Okay, that would also be dyslipidemia. So it's kind of a more broad umbrella term. One type of dyslipidemia is hyperlipidemia. That's what I'm going to be talking about today, okay? Let's take a look at some learning objectives. Let me see if I can get this. Hold on a second. Maybe not. Hold on. There we go. All right. So we're going to start off talking about the biochemistry and physiology of lipids. I don't necessarily think this is something you spend a whole lot of time with in biochemistry, okay? You know, I know you do talk about lipids and carbohydrates and proteins, but you might not talk about lipoproteins and how lipids are processed and managed by the body. So again, if you got done with a hypertension exam yesterday and you felt a little despondent for whatever reason and you went to the grocery store and got a pint of Ben & Jerry's ice cream, okay, which is something I do when I get despondent sometimes, okay? You know, how does your body process that? What's happening? What happens to all the fats in that particular type of food as it's being digested? We're going to talk about that today. Because I think, you know, we talk about lipoproteins. You've heard of some of these already. You know, you've heard the term LDL, right? HDL. You know, those two are very common types of lipoproteins that we talk about in a clinical setting. But you may not be able to visualize or understand what they are biochemically, okay? So we'll start with that. We're going to compare and contrast the structural characteristics. There's 5 different types of lipoproteins. They all are different in terms of their size as well as their composition, okay? And both of those are critical for determining their function. What do they do? Okay, so we're going to spend some time talking about that. We're going to talk about, that covers learning objective number 3. We're going to talk about the production, utilization, and elimination of the 5 types of lipoproteins. It's probably going to be a very basic conversation. I used to split this lecture up into 2 days. First day was all biochemistry, physiology of lipoprotein metabolism. Day 2 was the drugs. I've simplified it extensively here. So we're not going to get into that too much, but just enough so that you understand what's going on, okay? Then we're going to use all that good knowledge that you've developed about your lipoproteins to really begin to understand the mechanism of action of some of our drugs used to treat hyperlipidemia. Okay, so we'll use that as kind of our foundational scheme or framework in order to do that. We're going to talk about some structure-activity relationships, and there's going to be some med chem, okay? Not as much as with hypertension, but we're going to talk about specifically statins, okay, specifically. And then some of the side effects for each of the classes of drugs that we're going to see on the next slide, as well as their mechanism, okay? By far, the biggest class of drugs used to treat hyperlipidemia, and you all are familiar with these, are statins, okay? If you work at a community pharmacy, you're dispensing these all the time, okay? My drug of choice here is atorvastatin, okay? That's my drug of choice. But we have all the different lovastatin, fluvastatin, pravastatin, simvastatin, all the statin drugs. So again, you shouldn't miss that on your exam in terms of nomenclature. Statin, you know, these are our HM decoy reductase inhibitors, okay? We do have things like bile acid resonance and sequestrants. We'll talk a little bit about that. Inhibitors of cholesterol absorption. And then we have PCSK9 inhibitors, which are our newest class of drugs that are used in pretty extreme cases right now of hyperlipidemia. Dr. Naeem is going to talk all about the clinical guidelines and how they're used, but these drugs are very, very powerful and very, very effective, okay? All right, so starting off, we're going to start with the basics here and the fundamentals. What are lipoproteins, right? So when we look at that word, lipoprotein, there's 2 parts to it. Lipo obviously refers to lipids, protein referring to protein. So these are microscopic spheres or particles that are found in the body. A lot of times they are transported in the blood, so you'll find them a lot in the blood, that are made up of a variety of types of lipids, to include cholesterol and triglycerides, okay, as well as specific proteins, okay, called apoproteins. So we're going to talk about those and how they kind of contribute to the function of the lipoprotein. Right? Their job is to, again, transport these water-insoluble lipids. These are things that do not solubilize in the blood, okay, to target tissues. And those target tissues like skeletal muscle, the liver, adipose tissue can use them either for energy, those fats for energy. They can store them, okay? They can process them into something else, or they can eliminate them from the body. So there's a lot of different things that your body will do with these fats. I like to think of lipoproteins, okay, as sort of a bus, in the cardiovascular system that carries fats from one point to another. Okay, we're going to talk about why they're able to do that from a structural standpoint, and again, what kind of fats are each one of those lipoproteins composed of. Okay. As I said before, these are microscopic spherical particles. Again, these first two bullets you don't need to know. All right, this is just to impress somebody. Okay, if you're talking about lipoproteins, okay, you meet somebody that you're interested in and say, hey, did you know that lipoproteins are small spherical particles with a diameter of 7 to 100 nanometers, right? It was a miracle that I ever got married, right? It just, I don't know what Dr. Colleen was thinking, but at any rate, don't worry about that. It just gives you kind of an idea how tiny these particles are. Now, 3 major components to every lipoprotein we're going to talk about, okay? We're going to start first with the hydrophobic core. So at the center of the lipoprotein, the core of the lipoprotein, we have a hydrophobic core consisting of triglycerides and cholesterol esters. This is where the fat is that the lipoprotein transports. Okay? You all remember from biochemistry and SOAR, triglycerides are made up of a glycerol molecule and 3 fatty acids. You should remember that. The cholesterol molecule you can see over here on the right, or I'm sorry, on the left here, here's cholesterol. Again, it's a typical steroid-based structure, compound. Actually, cholesterol is used in the synthesis of a wide range of steroid-based hormones, so it's really important there. But it's also a type of fat that's found in foods, obviously, right? Now, the cholesterol, in order for it to be packaged inside of the lipoprotein, it's got to be slightly chemically modified. An ester group, it's a esterified, and converted to cholesterol ester. And you can see the ester group right here. This is the storage form of cholesterol in your lipoprotein. Okay? It just helps the lipoprotein to store the cholesterol, more efficiently in its core. Okay? Everybody got that? All right. Now, the second part of the lipoprotein is the phospholipid monolayer. All right? And I have monolayer in red there just to draw your attention. When we talk about like cells, we talk about a phospholipid bilayer, right, that makes up the cell membrane. This is a little bit different. This is a monolayer, just one single layer of phospholipids, okay? And you can take a look at it here. Here is a cartoon picture of a lipoprotein. And you can see here on the surface, okay, you've got these little lipoproteins here. If you see that there and there, the polar phosphate head faces outward, okay? And the fatty acid tails here face inward, okay? And the purpose of that phospholipid monolayer is to allow the lipoprotein to solubilize in the blood, because that polar head group has charge associated with it, okay? And you can see here, in this right picture here, you've got the hydrophobic core right in the middle here, okay? And you've got different things like cholesterol esters and triglycerides, okay, that are packaged inside of that and surrounded by that phospholipid monolayer. So that's part 2. Hydrophobic core containing fats, lipids. We've got a phospholipid monolayer surrounding that and allowing the particle to solubilize in the aqueous environment of the blood. Oh, what did I do here? Okay. And then we've got the third part, are apolipoproteins. Okay. I will tell you right now, I think the hardest part about this topic is all of the jargon, because all the words sound the same, right? Okay. Apolipoprotein, another term that's identical to this that I will probably use because it's easier to say, is apoprotein. Okay. All these proteins are proteins that are found in our lipoprotein. Okay? The hydrophobic core, right, is what makes up the lipo of lipoprotein, right? And then the apoprotein is what makes up the protein of the lipoprotein. Okay? Now, there are two types of apoproteins. We have integral apoproteins and peripheral apoproteins. Okay? I'm going to tell you the difference between the two of them. You need to know this. Okay? A integral apoprotein is a protein that the lipoprotein particle is built around. Okay? It's built around, it's an integral part of the structure of that lipoprotein. I want you guys to imagine, everybody here has seen a house being built, right? Being constructed, okay? It's like the wood framing on a house. You've ever seen a house framed up with just 2x4s and framing, right? With no walls or anything? And then all of a sudden, 3 days later, it's got siding on it and all that good stuff, right? So I like to think of our integral apoproteins just like the framing of a house. The house can't stand without it, right? It's there, it can't be removed, but it's part of the structure, okay? And the way that house frame is shaped, right? How many sides it has, how tall it is, right? How many rooms there are, will determine what the house looks like and how functional it is, right? And the integral apoproteins are the same way, okay? The lipoprotein is built around them, and it gives it its unique structural characteristics as well as some different functionality, right? Right? Now, peripheral apoproteins, okay, are apoproteins that are stuck to the outside of the lipoprotein, okay? All right? For example, okay, this might be a nice cartoon picture. It's kind of, it's the apoprotein here. It's kind of stuck to the outside of the lipoprotein there. Okay. These types of proteins can be added or taken away from the lipoprotein throughout its lifespan. Okay. And we're going to see how, for example, an LDL particle. All right, how its peripheral apoproteins change throughout its lifespan. All right, the analogy I like to use for this that helps people to understand that is pretend that you are a lipoprotein. Take your favorite lipoprotein, right? Maybe you're an HDL particle, maybe you're an LDL particle. Okay, every day you wake up, right? You go to your closet, you open it up, and you look at the clothes that you have to wear, right? And you say to yourself, "I don't have anything to wear today," right? At least if you're like my kids, okay? All right? Or if you're like me, it's like, I don't know what tie to wear today. What tie goes with what anyway. But you choose, a lot of times you choose the clothes, the type of clothes that you're going to wear based upon the weather and what you're going to be doing, right? If you're coming to Care Lab, right, you might dress differently, choose to dress a little bit more professional, right, for Care Lab or to go visit your patient, than if you were going to the gym later on, right? Different clothes for different environments and different functions. Think of these peripheral apoproteins as clothing that your lipoprotein can put on and take off as it's needed, right? Okay? If you came in here and it was 35 degrees in the Knott Auditorium and you had to sit here for class, right? You'd probably put your coat on, right? To stay warm. But when you go outside, maybe it's 70 degrees out there. You're going to take your coat off because you don't need it, right? That's what happens with our peripheral apoproteins, all right? Lipoproteins will pick them up. They'll get stuck on them. They use them for different things, and then they get rid of them when they don't need them anymore. Very dynamic system here, all right? Any questions about the difference between these two types of proteins? One's like the framing of a house. The other one is like clothing you pick out of your closet to wear. All right, to adapt to different situations and to do different things. All right? This is my own cartoon picture here of the three components of an lipoprotein. Okay, so again, at the core here, we have this red core center. That is our hydrophobic core, right? And hydrophobic means water-hating. And it contains things like cholesterol esters and triglycerides. And again, depending on what lipoprotein we're talking about, it may have more or less of these. We'll get into that in a minute, okay? Surrounding the hydrophobic core is our phospholipid monolayer. This is what allows the lipoprotein to solubilize in the blood. The hydrophobic core by itself can't because it's hydrophobic, right? It's made up of lipids, so it don't dissolve in water, okay? But that phospholipid monolayer allows for the particle, because of its charged properties, will allow for dissolution in the blood. We have our integral membrane apoproteins. Okay, right here, these are like the framing of a house. The lipoprotein is built around these, and it stays with the lipoprotein throughout its entire lifespan. And then we have a peripheral membrane apoproteins that, again, can be stuck on the surface of the lipoprotein, allowing it to do certain things, and then it can be removed when it's no longer needed. Okay? Everybody got that? All right. So far, so good. All right. So in terms of how we classify our lipoproteins, like I said before, there are 5 different types that we're going to talk about. And they are typically classified either in one of 2 ways, either based upon their physical characteristics or their functional characteristics. Okay? So each one of these lipoproteins have a different density, a different size, and a different protein content. That's more of a way to structurally classify them based upon their density, size, and protein content. Typically, we use that in combination with functional classification. What does that lipoprotein do in the body? For example, we have our VLDLs and chylomicrons. Okay, these particles, as you're going to find out shortly, are critical for transporting triglycerides throughout the body. Okay, one of them is produced during fasting. The other one is produced after eating a high-fat meal. So again, last night, if anybody here had that kind of Ben & Jerry's, okay, you were ingesting a lot of triglycerides, and your body was adjusting to that by producing a lipoprotein that would carry it to get stored somewhere. Maybe you didn't want it stored, like right here. Okay. We have cholesterol transport. Okay. High-density lipoproteins are really important for transporting cholesterol through the body. One thing I want to really clear up with everybody is when we hear the word cholesterol or triglycerides, what do we think? Fats, right? We think it's bad or good? Bad, right? Everybody wants to avoid cholesterol and triglycerides, but does your body need those things?
Yes.
Absolutely it does. Okay? It's everything in moderation, as Benjamin Franklin said. Okay, and probably the ancient Greeks said that way before him, but at any rate, everything in moderation. Lipids, like triglycerides, cholesterol, super important for body functions. It's just when the regulation of their metabolism and the way that they're handled goes awry, that's when we can run into problems. Or if we have a diet where we consume too much of them. Okay? HDL sometimes is known as the good cholesterol. Right? There is no good or bad cholesterol. If it helps you to remember this is why and sometimes they refer to it as the good cholesterol, because HDLs are known for scrubbing or eliminating excess cholesterol from the body. And so again, if you look at a lipid panel, you want your HDL levels to be high, but again, they can get too high, right? But high enough where the HDLs can help to remove excess cholesterol from the body. That's not a bad thing, that's a good thing, okay? The other thing that HDL particles do, you're going to find out, is they serve as a repository for peripheral apoproteins, the things that we can stick on the surface of our lipoprotein. So I like to think about HDL as sort of like the closet in your bedroom, right, that stores your clothes. The clothes are your peripheral apoprotein. You can put them on and take them off. Closet, or dresser, or whatever, is what stores them, okay? And that's where you go to get them. And that's where you go to put them when you're done with them, right? Okay? Unless you're like my son who likes to just toss them on the floor. Okay? All right. But at any rate, that is, that's what we're talking about here. All right, so let's get into it. 5 types. The first type we're going to talk about are chylomicrons, abbreviated CM. You probably have never heard of these before. Okay? These are, but they are the largest of the lipoproteins. Okay? But they're the least dense. Okay, so one of the things I'm going to tell you is that as we go through these lipoproteins, they're going to get smaller and smaller in size, but their density is going to increase. Okay? They're going to get more dense as they get smaller, right? Because of their composition. Now, chylomicrons are what's made in your, in your body after fat is digested and absorbed. Okay? So chylomicrons, like let's say you sit down and you ate that pint of Ben and Jerry's, your body's making tons of chylomicrons and then taking the triglycerides and fats in that food, that ice cream that you just digested, and transporting it to target tissues in the body, like muscle tissue, like liver tissue, like adipose tissue, right? And then that tissue can do a lot of different things with it. They can use it for energy, they can store it, there's a whole a litany of things that can happen, all right? The pathway name for this, when chylomicrons are produced, it's called the exogenous pathway of lipoprotein metabolism. Exogenous means outside of the body. So this particular pathway where chylomicrons are produced, they're produced because of ingestion of fats from outside of the body, okay? Right? So again, anytime you sit down and eat your Ben and Jerry's, just think you're making chylomicrons, okay? Because your body has to process that and store it and use it. Okay? Now, composition here. The vast majority of the weight of this lipoprotein comes from fats, okay? So 98% of the weight of this particular particle is because of fats. And mostly important here is that it's largely triglycerides. So chylomicrons are critical for transporting triglycerides, from the GI tract to different target tissues in the body, okay? Now, there are a couple of different apolipoproteins, again, also known as apoproteins, okay, that are associated with chylomicrons, okay? The first one here is ApoB48. This is the integral protein that your chylomicron is built around. So this is like the framing that your chylomicron is built around. So you have enterocytes in your small intestines, okay? They make this protein, ApoB48. And as you ingest fats, fatty foods, it will take that fat and start to construct a chylomicron around this protein. Everybody following me? Okay? And intestinal cells, are very unique. They're the only ones that are that have the genes to make this particular protein. Okay? So very specialized. Everybody understand that? Then we have these two. There are a lot of other ones, but I wanted to try to keep it as simple as possible. We have ApoC2 and ApoE. These are peripheral apoproteins. They're added on to the lipoprotein during different phases of its metabolic lifespan. And for example, ApoC2, when it's attached to the surface of your chylomicron, allows that chylomicron to activate a specific enzyme called lipoprotein lipase. Okay? And this enzyme actually helps to unpack the chylomicron, remove some of its triglycerides from its hydrophobic core. Okay? It's very important. Okay? ApoE is another peripheral apoprotein that is added to our chylomicron during its metabolic lifespan. And this particular apoprotein allows the chylomicron to be taken up by the liver. Okay? So these peripheral apoproteins like ApoC2 and ApoE act as ligands. Ligands for different enzymes or receptors that allow the chylomicron or HDL particle or LDL particle to do something specific in the body. Everybody got that? And without it, they can't do it. Okay? Everybody got it? All right, good. The next one, all right, are very low density lipoproteins, abbreviated VLDL. Okay? So these are the next largest in size of our lipoproteins. And, okay, they are low density. All right, very low density lipoprotein, VLDL. They do have a low density, but the density is slightly more than the chylomicron. Remember, I said these lipoproteins, as we talk about them, they're going to get smaller and smaller and smaller, but they are going to get more and more dense as well. Okay? VLDL particles are made in the liver, okay, during periods of fasting. Okay, where lipids aren't actively being digested or consumed. Okay? And so the VLDL particle actually takes triglycerides that the liver makes, okay, and transports those triglycerides again to target tissues of the body like liver tissue, like liver, adipose tissue, skeletal muscle, et cetera, okay? So very similar to chylomicrons, it's just the source of triglycerides are different. We refer to the production of VLDL particles as the endogenous, okay, pathway, metabolic pathway here, or endogenous pathway of lipoprotein metabolism, because the triglyceride source, the source of triglycerides that the VLDL particle is made of, is made inside the body. It doesn't come from stuff. It doesn't come from dietary foods from outside the body. It comes from the liver, and it's triglycerides that the liver makes. Okay. So again, chylomicrons, VLDL particles, very, very similar. You know, chylomicrons are used to transport exogenous fats that we take in through our diet. VLDL particles are used to transport triglycerides that your body needs, specifically your liver. Right? Again, I said the main job here again is to carry triglycerides from the liver to target tissues. So very, very similar to our chylomicrons. They are, again, about 90% lipid by weight, so they have a little less lipid in them than the chylomicrons do. Okay, not much, but a little bit. And you can see here that 50% of the fat content is triglycerides. Okay, so again, the amount of triglycerides carried by VLDL particles is less than chylomicrons, but it's still a critical mechanism for transporting triglycerides made by the liver. Okay, now in terms of apolipoproteins, we have our ApoB100. Again, this is an integral apoprotein, meaning that it's the framework around which your VLDL particle is built. All right? ApoB100 is only made by hepatocytes or liver cells. All right? That's the only cell type in the body that I'm aware of that has the genes to make this particular protein. And it's very, very similar to ApoB48. Okay, only it's bigger. All right? So the ApoB48 is actually a smaller version of this ApoB100 protein that's made in the liver. They're related. One's just smaller than the other. They have the same amino acid sequence up to a point, and the ApoB48, again, has less amino acids than the ApoB100. And then we have the two apolipoproteins that are added peripherally to the VLDL particle throughout its metabolic lifespan. We've got ApoC2 and ApoE. Again, very similar in terms of function, allow for activation of lipoprotein lipase, okay, as well as being taken up and cleared from the body by the liver, okay? So that's important, all right? So and there are many more peripheral apoproteins that are associated with this particle, but we're not going to get that deep into it. We don't need to get that deep into that, okay? Any questions about VLDL particles? All right. So again, what's really important is to be able to kind of compare and contrast chylomicrons and VLDL particles in terms of their function and their structure. Next, intermediate density lipoproteins, or IDLs. Okay, these ones, again, are probably lipoproteins that you're not familiar with, okay? As their name implies, their size is intermediate, okay, because this is kind of the middle lipoprotein. Anybody the middle child in their family, right? This is kind of the middle child of the lipoprotein family, okay? You can see here that in terms of their formation, these lipoproteins are remnants of VLDL metabolism. So what happens is during the lifespan of our VLDL particle, the life of the VLDL particle begins in the liver, as the liver is making triglycerides that need to be transported throughout the body. The VLDL lipoprotein is produced then and released from the liver into the bloodstream. It starts to float around and it comes into contact with a target tissue that needs triglycerides. And it docks with that target tissue, okay, through interaction with lipoprotein lipase. And that lipoprotein lipase starts to empty the hydrophobic core of triglycerides. And as that happens, the VLDL particle begins to shrink, and its fat composition begins to change, and eventually it turns into an IDL particle, okay? I like to think of IDLs, okay, as a VLDL particle that lost weight, okay? Lots of weight, lost a lot of weight, and it's now a different particle, okay? If that will help you to remember it, great, okay? Again, once the IDL particles are produced, half of them get eliminated directly from the body. The other half are converted into a lipoprotein that we're going to talk about next, which is your LDL particles. Okay? So VLDLs, IDLs, and LDL lipoproteins are all related to one another. They're all essentially the same particle, but just different sizes and composition. Okay? One is made as the other one is metabolized and changed. Okay? High lipid content here, 82%. So a lot of lipids in your IDL particle. And again, we've got the same integral apoprotein here, the same framework, the ApoB100 protein, as your VLDL, right? Because it's the same particle, just less lipids on it, okay? And ApoC and ApoE are also peripheral apoproteins that, again, are important for the function of the IDL particle. All right, so we're kind of in the middle here. IDLs are produced as VLDL particles are being metabolized and changed. Half of them get eliminated from the body via the liver. The other half are converted into our next lipoprotein, which is our low-density lipoprotein, LDLs. Okay, we often think of LDLs as what type of cholesterol? The bad cholesterol, right? We don't want high LDL levels, right? Okay. Again, there are no good or bad types of cholesterol, but this type of cholesterol or lipoprotein is associated with atherosclerotic disease. Okay, we're going to talk about that next when we talk about coronary artery disease. Right? The thing about atherosclerosis is, in the formation of plaques in your arteries, like your coronary arteries or throughout your body, we've learned a lot about the process in which those plaques form and what are the factors involved in that, but we don't know everything about it yet. Still a lot of question marks there, okay? But we do know that, again, LDL particles, especially when there is an excess of them, can undergo chemical changes that allow them to be incorporated into atherosclerotic plaques. Okay, these are, again, your plaques in your blood vessels that can cause coronary artery disease or peripheral artery disease, okay? So again, how are LDL particles produced? Well, they're produced when IDL particles are metabolized, okay, and altered in terms of their structure and their composition, okay? And the purpose of this, of the LDL particle, is to carry cholesterol, okay, to target tissues. Right? So what happens here, and I'll let you, if you're going to take notes on that, what really happens is you start off with a VLDL particle that's maybe in the liver. Okay, it's full of triglycerides and a little bit of cholesterol in the hydrophobic core. As it goes through its metabolic lifespan, it'll start to lose triglycerides from the hydrophobic core, and it becomes smaller and more dense. Okay, and it becomes an IDL particle. And then that IDL particle, again, as it is metabolized and goes through its lifespan, it loses even more triglycerides. So that finally, when you get to an LDL particle, the hydrophobic core is full of cholesterol. There is still some triglycerides there, but there's a lot of cholesterol there. And these tiny, dense particles are what carry cholesterol throughout the body. Okay, and again, your cells use cholesterol for cell wall synthesis, for hormone synthesis, and a whole bunch of other things. Okay, so it is important. It's just when there's too much of it, that's when we've got a problem. Okay. It's very small in terms of its protein composition. Again, it's got a lot of cholesterol in its hydrophobic core, and it contains only the ApoB100 protein. That's the only kind of protein that it typically is associated with. And that's the same ApoB100 protein that the VLDL particle that it started as was built around. Okay? Some kind of clinical implications here, all right? There is a variant of the LDL particle or lipoprotein called LPA, all right? And essentially this is an LDL particle with an ApoA peripheral protein attached to its outer surface. This type of LDL particle is highly associated with atherosclerotic disease, okay? It can, again, interact with foam cells and participate in the formation of atherosclerotic plaques. So again, there's a lot of research going into this different type of LDL particle. It's kind of unique, and its role in atherosclerotic disease. Okay? Everybody got that? And then we have our last one, our HDL particles, right? Our HDL lipoproteins. These are the smallest but most dense of the lipoproteins. Okay? That's because in their density, their high density comes from all the protein that they carry. Okay? HDL particles are produced in the liver. Right? When you take a look at their composition or content, they've got the lowest lipid content of any of our lipoproteins, but the highest protein content. And this composition or content really allows the HDL particle to do the types of things that I talked about at the beginning of the class. HDL particles do two really important things. Number one, they're cholesterol scrubbers. Okay? They remove excess cholesterol from target tissues. Okay? They don't have a lot of fat or lipids in their hydrophobic core, so you can think about it in this way that they have room for some extra fat there. Okay? And so they'll take extra cholesterol from target tissues, package it up inside its hydrophobic core, and then it can get eliminated from the body. And that's how your body can deal with excess cholesterol. Okay? It also serves as the repository or closet for our peripheral apoproteins. Remember, those are your clothes that you put on every day to allow you to adjust to your environment and do specific activities. Okay? All right? So HDL is sometimes referred to as good cholesterol. This is the type of lipoprotein where you'd like the levels a little bit higher, right? But again, like with anything, they can get too high and it can be a problem, all right? So that's that there, all right? Okay. This table here, okay, is just FYI, all right? So if you weren't following along before or you're having some trouble with that, take a look at this table. All it shows you here are the different types, okay, of things that the lipoproteins are made of. Okay, protein, triglycerides, cholesterol, cholesterol ester, phospholipids, and apoprotein. And then here are our 5 lipoproteins: chylomicrons, VLDLs, IDLs, LDLs, and HDLs. Again, you can see as we go down this list, the protein content continues to increase. Okay, the triglyceride content will decrease. Okay, and the cholesterol content, gets higher as you move down here as well too. So there's some trends to look at. If you understand what these lipoproteins are made of, you can better remember and understand what their function is in the body. All right? Any questions? Nothing. This is post-exam 1 destitution. Is what we're experiencing. Right? You'll be swept in a week, right? Okay? Everybody good? Any questions? No? We don't care? Yeah, you will care when you get to be my age and you got hyperlipidemia. You really want to know about that, okay? All right. Okay. So, some study stop questions. I didn't have these in hypertension, but I do have these here. Your study stop questions are positioned at strategic locations throughout your lecture, okay? And again, as you're studying, you can stop and see if you can answer some of these questions. They're pretty basic. They're recall questions, but at least it gives you an understanding of, hey, are you remembering this? Can you explain basic concepts? And it can help you to prepare for the exam. So don't skip over these. These are really important, okay? All right. So let's talk a little bit about how your cells kind of process cholesterol, okay? Most of your cells, okay, in your body can make cholesterol. They have the enzymes that are necessary to make cholesterol. We'll talk about that in a minute. But they also use cholesterol that comes from exogenous or external sources. So again, here we have a picture of a target cell, okay? Let's pretend that this is a liver cell or hepatocyte, okay? And we can see up here, we've got an LDL particle, okay? We know that because, again, it contains a lot of cholesterol in its hydrophobic core, and it's built around this ApoB100 protein. Okay? Remember, LDL particles are responsible for transporting cholesterol throughout the body. So here we have one transporting cholesterol to this particular cell. Okay? Now, what happens here is that the LDL particle can interact with special receptors on the surface of our hepatocytes and other target cells called LDL receptors. Okay? And what you can see here is that as that happens, okay, phagocytosis occurs and the cell takes up the entire lipoprotein particle and pulls it inside of itself. Okay? And that can fuse, okay, with a lysosome, which contains digestive enzymes, essentially, right, that break down protein. And what we get here is that the LDL particle is slowly broken apart or dissolved, and its core substances are now released into the cell. We've got amino acids here from our ApoB100 protein, but more importantly, what's released are your cholesterols from the hydrophobic core. You can see that right here, okay? And that cholesterol is then, again, utilized by the endoplasmic reticulum. And what ultimately happens here is that the cellular machinery, once your cell takes up some LDL particles, the cellular machinery that's either responsible for making new cholesterol, okay, or, okay, for capturing more LDL particles, their activity changes. So again, we see the uptake here of an LDL particle. All the stuff that the LDL particle carries is now part of the cell. It's like going to the grocery store and filling up the refrigerator and the cabinets, right? Now your refrigerator's full, your cabinet's full. You don't need any more food. You don't need to grow food at home. You don't need to go back to the store. Everything's full up. So what happens is you get a reduction in the activity of HMG-CoA reductase. This is one of the key enzymes involved in cholesterol biosynthesis. You've seen this term before because guess what? This is the enzyme that what group of drugs target? Statins, yes, okay? So this is a key enzyme in the production of the intracellular production of cholesterol, okay? And its activity goes way, way down after a liver cell picks up an LDL particle. Why would the cell need to make cholesterol if it just picked up a bunch of it from an LDL particle? It's like getting Uber Eats, right? Something delivered, right? So you're getting some food. You don't need to go out and buy food. You don't need to make it. You've already got it, okay? ACAT, okay? The activity of this receptor, okay, or this enzyme goes up, okay? ACAT is responsible for converting cholesterol esters, okay, to the storage form of cholesterol. All right, so it's kind of an enzyme that's involved in processing cholesterol that the body's just taking in. Bless you. And then we have a reduction in cell surface LDL receptors on this particular cell, right? The cell's captured a bunch of LDL particles. It's full of cholesterol. It's full of amino acids. It doesn't need to collect anymore. So what is it going to do? It's going to reduce the number of LDL receptors on the surface of the cell so that the cell doesn't capture anything, okay? So what I want you to get at this, from this, is that lipid metabolism is very, very dynamic inside of cells, okay? And cells can make their own lipids, but they also get lipids from our lipoproteins. And again, that can also change the activity of the cell and how it's processing lipids, all right? This is going to become really important when we talk about how some of our drugs work later on, okay? Any questions about this? Any questions? Okay. as I said before, all of your cells in the body, most of them at least, can have the ability to make their own cholesterol. They don't need to get it from LDL particles. They can make it themselves, okay? But I think it's a complex process. It involves a lot of different enzymes, okay? It's probably very energy intensive for the cell. Let's take a look at that process right here, okay? So here we have the beginning molecules in terms of cholesterol biosynthesis, which you see right down here, okay, is acetyl-CoA and acetoacetyl-CoA, all right? And these two substrates interact with HMG-CoA and HMG-CoA reductase. Well, they're converted to HMG-CoA, and then HMG-CoA reductase converts that particle to mevalonate, okay, which is a molecule that's part of the cholesterol biosynthesis process. You can see here, look how many steps there are, okay? And these are all cholesterol intermediates, okay? There's a ton of them. Now, I expect you to memorize all these for the exam, okay? All right? So make sure you know all these and how to spell them, all right? No, I'm just kidding. Come on, guys. Whatever. I saw you writing it down. I'm like, oh my gosh, I'm scared. No, you don't need to know that. Jeez. Okay, I'm so sorry. You guys, I feel bad. You're not. It's like, are you still there? Are you alive? I'm feeling kind of bad today. All right? You got to help me out here, okay? You fake it. Just fake it. All right? If you're falling asleep, paint some eyeballs on your eyelids or whatever, okay? Right? That'll work or whatever it takes, okay? What do you need to know, though? Let me show you what you need to know. Right here. See this right here? HMG-CoA reductase. This is the second enzyme in the cholesterol biosynthesis process. This is your key target for your statins. You've got to know that. So I want you to know, A, that HMG-CoA reductase is a critical enzyme in the biosynthesis of cholesterol. Okay? I don't even care if you know what its substrate or product is. Just remember that, okay? And that it is the primary target for your statin drugs. Okay? Statins, I'll let you guys finish writing that down. Okay? This may be a surprise to all of you. Okay? Statins really don't do anything to the cholesterol that you take in the food you eat. Okay? And that's a big misconception that people have. Like, well, I'm taking a statin so I can eat whatever I want, right? It's for, right? No, it's not the way that it works. And I'll be honest, sometimes it's the way I think it works, right? Even though I know better and I teach this, okay? All right? So when I sit down and eat, at least I feel guilt when I sit down and eat that Ben & Jerry's ice cream, right? Okay? Statins, all they do is inhibit, and it's not, I don't mean to say that it's all they do, right? It's important. They inhibit cellular biosynthesis of cholesterol. So your body's got two sources of cholesterol, from the foods that you eat and from the cells in your body, okay? And those together make up the total cholesterol inside your body. So again, what we're doing here is we're targeting the body's ability to make cholesterol. You're still getting stuff from your diet. So if you're taking a statin and you're having a pint of Ben & Jerry's every single night, your cholesterol is probably not getting any better, okay? But if you're following a reasonable diet, you're exercising, and you're taking your statin, that can help lower your cholesterol, okay? Yes, sir?
That was my question.
Yeah.
That, like, if you take a statin but still, like, eat pizza every day,
Yeah.
whatever.
Probably not going to help out too much,
Okay.
right? Yeah. I mean, and again, so here's the other thing, too, I want you guys to understand. Some people, because of their genetics, okay, because, dyslipidemia is something that, again, there's a genetic component to it, okay? How many people have dyslipidemia in their families, right? All right? And I do too. My whole family has it. So I wasn't surprised when I got it. But at any rate, some people overproduce cholesterol in their cells. And so statins can be super, helpful. You might have the healthiest diet on the planet, and you still have high cholesterol, okay? And it's not because of the food you're eating, it's because this process here is dysregulated in some way. Does that make sense? Okay? Now, okay? Let me also kind of show you, and I'm going to come back to this picture when I come to the statins again, because there's a bunch of stuff down here. You look down here at the bottom. I just want to show you really quick. If we have a statin on board and we inhibit this enzyme, what happens to cholesterol production in the cell? Does it go up or down? It decreases, right? It goes down. And the cell will respond to that, okay? And this is the process by which the cell responds to that decline in cholesterol production. And it involves making more LDL receptors, right? Okay. So that the cell can now capture more cholesterol, because the cell likes to have cholesterol, okay? So you can even inhibit that process, right, of intracellular cholesterol, okay? But the cell will make, okay, more LDL receptors, okay? And what happens as a result is that these cells start taking up LDL out of the bloodstream and removing it from there, okay? So not only are we inhibiting, okay, the cellular production of cholesterol, but we're also helping the body clear out excess LDL particles from the blood, okay? So that's important, okay? All right. One of the things that I want to point out here, when we talk about LDL receptor regulation, because this part right here is a key component to a lot of the activity of some of our major drugs right here, okay? All right, like our statins, what you're doing is you're inhibiting cholesterol production inside the cells. The cell responds to that by increasing LDL receptor production, and now the cell will start pulling out lots of LDL particles out of the bloodstream, lowering plasma LDL levels, which is good, okay? The heck am I doing here? All right. There's an important protein here, okay, that is involved in regulating this process. Okay, and this is called PCSK9, okay? And what this particular protein does, okay, as you can see here, over here, well, we'll start here on the left here. All right, so here we have another liver cell, for example, okay? And you can see here it's got these LDL receptors on its surface. And we have this LDL particle that's floating along the bloodstream or in the extracellular fluid, and it attaches itself to the LDL receptor, okay? We've seen a picture of this before, just in a different way. The LDL particles are pulled into the cell via endocytosis. It is packaged in a clathrin-coated vesicle, and then it's broken down. And as it's broken down, the LDL receptor that originally caught that particle is recycled and reinserted into the cell membrane. That's kind of the normal process here, okay? The PCSK9 protein, okay, is a protein that's produced and released by this cell, and it helps to regulate this process over here, okay? So let's say we have an example where this tiny little hepatocyte here is making and releasing PCSK9, and that PCSK9 protein starts to bind to LDL receptors, all right? And that LDL receptor picks up an LDL particle, it internalizes it via endocytosis, okay? But you can see here, look, there's the PCSK9 protein right there. It looks like that kidney bean, okay? And what happens is, instead of the LDL receptor being reinserted into the cell membrane, as you see over here, it's broken down inside the cell, okay? And what that does over time is it lowers the LDL receptors on the surface of cells throughout the body. And so your body can't appropriately clear LDL particles out of the bloodstream if this process goes awry. Again, there are certain people, all right, that have a genetic predisposition. They make an excessive amount of PCSK9 protein, okay? Think about it. Think if you're a patient or person and your body is overproducing PCSK9 protein. And what happens is over time, a lot of the cells in your body lose the LDL receptor. Can those cells now capture and remove LDL particles out of the bloodstream? No. So what happens is the LDL stay in the bloodstream, okay? And their levels increase. So these are people that have excessively high LDL levels, okay? Yeah.
Is that like is that one of the mechanisms of familial hypercholesterol?
It can be, yes. But and this is really, and what you'll see, and a lot of times in patients that have like wildly bad cholesterol, like super high, abnormally high levels of LDL, not all the time, but a lot of times this can be part of the process. And the new drugs, the PCSK9 inhibitors, they're amazing at dealing with this problem, okay? We'll talk about them later on, but just if you're curious, right? PCSK9 inhibitors are monoclonal antibodies that will bind to the PCSK9 protein and shut it down. And all of a sudden now, your cells start producing and inserting LDL receptors into the cell membrane, and your cells regain the ability to capture LDL from the bloodstream. There's some remarkable results with this, okay?
Is this new treatment?
These are new to me, probably new to you. They've been around, I would say, if I can remember, it's probably about 10 years ago, 8 to 10 years ago when I started teaching this. I'm ballparking, okay? But of all these drugs, they're the very newest class, okay? And there's a lot of different examples out there now, that are available. But again, they are really amazing drugs with regards to being able to deal with this kind of problem, okay? This is a great example of, once we learned how the LDL receptors on cells were regulated, and we discovered that PCSK9 protein, we were able to develop a drug to target it, and it worked really well, okay? So, good stuff. All right, we are at a perfect time for the break, okay? And then we will get into the drug molecules, but only after I take your questions when you come back, okay? Let's see if we got any questions when you come back. All right? Let's take 6 or 7 minutes, and then we'll return for part 2.
I was like, no. Sorry. Can you do it? Okay. I didn't even
I was looking at it yesterday. I was like, ew.
You're really very well. You feel it better?
Better, but are still.
Yeah, but
it could be way worse. I mean, I might have my fix sometime, maybe next week. But I don't know. It doesn't really.
at least you have a I have friends. I have one friend that fell off and broke his like he has to use his retainers. Double this, yeah.
Yeah, I need to. It's wait for it.
No, you can fix.
I mean, the lips part, of course.
The thing, yeah. I get it.
Like that's. I want to say that's. And I literally tripped on like head damage.
Yeah, but
It happens, yeah.
so do you have plans for today? What you gonna do?
So later we're gonna go to you know what Hawkhead is?
No.
Like the shoes, the orthopedic shoes, the nice ones. We're gonna go to tour it and like clothes and stuff. And then after that,
Oh, nice.
how about you? You're all dressed up.
Oh, really?
Oh no.
3:30.
Oh, and she
Yeah.
oh, kidding! I didn't like that.
What?
You could
Oh, okay.
Like, have someone take a picture, you guys. And send it to me so I can put it in my or Jessica is like, basta, video, take a picture, please. And like post it. Oh, okay. Yeah. So see. Like, I can send me a picture. You can post it already.
The room was like, I need that. Like say, in the Zoom. I don't know what. Yeah. I was just falling asleep. I was like, Dr. Coach, I need to go back to the lecture and redo everything again because I didn't understand anything.
She was texting me.
Right, if I knew I would be like, I'm like, just like, drop it like, yeah. Going down the street. It's a huge honor. Yeah, everyone that I wait and like some people are like, yeah, okay, it's a good fight to church.
Yeah, it's like, no, that you have to like maybe good grades or something.
Yeah, that's
That's what it is.
This is really good as you're smart.
Yeah. Oh, they're gonna have a program with them.
Dr. Cohen, in the process of the recycling of the for the LDL receptor, is like there an equilibrium between the PCSK9
So, that process is working on the PCSK9
it's like is the transcription mediated?
It is, absolutely. So, just any other protein, transcription translation. And it's a normal part of this process, but when too much is needed, that's when your cells usually are going to take LDL out of circulation. And the receptors like them. So it stays in the blood.
Okay, and then they come up
Yeah, there you go. Yeah, we're going to talk all about that.
Yeah, okay.
2-minute warning, 2-minute warning. We're all back? No? You get 2 minutes, you can talk. It's tough in here. It doesn't feel like there's any AC. It's very warm in here. I know. I feel for you guys. You know what's so frustrating? When I was a grad student, when I was here, we used to have these Monday afternoons at 3 o'clock seminar. So we'd have external speakers come in, or people from the medical center come in. And there was always a run for the coffee pot in the office, like between 2:30 and 3. You'd get down there and get coffee. And it was awful, like my father, my AP yesterday, one guy, he fell asleep and fell off his chair.
Oh my
in the middle of the
God.
auditorium. Just fell off. And it was so embarrassing. And he just felt like life. And I would doze off for a minute or two. It was so frustrating because then I had a question I wanted to ask, but I was too embarrassed to ask it because I thought while I was asleep that they had gone up and missed it, right? And I didn't want to look a total idiot in front of everybody. So there were a lot of questions that didn't get answered, I guess. But so I can appreciate that for sure. I appreciate you guys hanging on.
Yeah, because like after lunch,
Oh, it's terrible.
we had an exam yesterday.
Oh, it's terrible. And it's hot in here.
It's
Warm.
hard today. Yeah.
I feel like everything was.
I could do magic tricks up there. I don't care. Right? Well, that's at least not funny. Yeah. All right, you guys ready? Don't answer that question. We are making good progress. But I don't want to make it at the expense of not understanding what we're getting at. So just to recap real quick. We talked about the general characteristics of lipoproteins, right? These are lipid protein particles, small spherical particles, whose main job is to transport water-insoluble lipids throughout the body, right? They're like the tractor trailers, right? The semi-trucks of the body that carry lipids from one part to another. So maybe scratch the bus analogy and think about them as tractor trailers, the semi-trucks on the highway. They're loaded up with lipids. They carry from one part of the body to the other so that target tissues can store them, use them for energy, use them for synthesis of other molecules, all that good stuff, okay? There are there's 3 different parts to them, right? We got the hydrophobic core where all the fats are. It's like the trailer on the back of the semi-truck. We've got a phospholipid monolayer surrounding the lipids and allowing them to be solubilized in blood. And then we have our protein component. There are 2 types of proteins we've kind of talked about. The integral apoprotein, that's analogous to sort of the wood framing on a house that the house is built around. Same thing with that. There are 2 major types of integral membrane proteins, ApoB48, produced by enterocytes in the small intestines, and then we have ApoB100, which is produced by liver cells. Okay? The second type of protein associated with these particles are apoproteins. These are like clothes in your closet. They can be added to the lipoprotein and taken away from it during its metabolic lifespan. And these apoproteins allow the lipoprotein particles to do lots of different things in the body which are important. Okay? Just like, again, if you wanted to go to the gym after class, you're not going to wear the clothes you wear right now, right? We're going to put your gym clothes on and your sneakers, and that allows you to do what you need to do. But when you're done, right, come back, you put those clothes away, okay? Hopefully in the washing, right? Okay, not back in the closet. And then you put other clothes on, all right? So to do something else. So very, very important. Each of the different lipoproteins, chylomicrons, VLDLs, IDLs, which remember is like the middle child, LDLs and HDLs, okay? These are our 5 major types of lipoproteins. They all differ in size, density, and composition. Okay, protein composition and lipid composition. Their lipid composition will tell you an awful lot about what they do. Okay, for example, chylomicrons and VLDL particles have a ton of triglycerides in their hydrophobic core. That's what they carry. Okay, that's what they're loaded up with. And they carry triglycerides from one part of the body, either the liver where they're made, in the case of VLDLs, or from the intestines, okay, where you're digesting and processing those triglycerides that you ingested from your Ben & Jerry's ice cream. Okay? And they get transported all over the place. VLDL particles, as they go through their metabolic lifespan, and triglycerides are removed from their hydrophobic core, they get a little bit smaller, they get a little bit more dense, and they ultimately become intermediate density lipoproteins, IDLs. The IDLs are either removed from the body or they're converted to the next lipoprotein, which is LDL. Okay? And they're converted to LDLs by further removal of triglycerides from their hydrophobic core. So when you get to the LDL particle, the vast majority of lipids contained in its hydrophobic core is cholesterol. And that's what LDL particles do. They transport cholesterol from one part of the body to the other. And there's a variant of LDL particles, okay, LPA that we talked about. This is a type of LDL particle that's got an apoprotein on it that it probably shouldn't. That is essentially thought to be a major component of the formation of atherosclerotic plaques. We'll talk about that next. Okay, in the next series of instruction, on the next topic. So we talked a lot about that. We talked about how cells can either make their own cholesterol through the cholesterol biosynthesis pathway, or they can grab cholesterol from the bloodstream by binding to LDL particles and pulling them out of the blood. Okay? And again, both processes are important. But we also talked about a situation where in certain individuals, a protein called PCSK9, which regulates the LDL receptor cycle and population, okay, can be overproduced. And those people generally lose a lot of ability to actually extract LDL particles from the bloodstream and have them taken up by target cells. So their LDL levels go way, 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 way up. Right? And we have a specific type of drug that targets that protein, okay, and turns it off essentially, allowing cells to restore their ability to capture LDL particles out of the bloodstream and lower LDL levels. Okay, so that's kind of the recap. Any questions about any of that? No? While you're thinking or asking questions, I'll get a drink of Mountain Dew here. Kingsley, you had something?
Yeah, I'm looking at this last part of it, the PCSK9. They're naturally present in the body.
They are. They're naturally present in the body.
So is there a situation whereby people do not have it?
There could be.
Deficient in it and whatever?
I'm not aware of that, but it's not necessarily my field of research. So there could definitely be a situation where the patient said, don't make it, right? And then what happens? Then you've got a ton of LDL particles that are being taken up in the cells at very low LDL levels, which may not be good either. Because again, LDL particles by themselves aren't bad, right? They transport cholesterol, right? And your body needs cholesterol for a lot of different things. So that in itself can be, but what we typically see in these, and I think it's one of the ways we kind of discovered this, is there's a certain subpopulation of patients with hyperlipidemia that have like outrageous LDL levels. I mean, like super high, more than what even someone like myself has. I have hyperlipidemia too, right? And so, by studying those folks, they discovered, okay, that an excess of this protein was being produced, and that's why the body couldn't process those LDL particles. So they developed a drug to inhibit a monoclonal antibody. Okay, so it's just an interesting, Yeah, it really tells a great story about how the more we understand disease processes and pathophysiology, the more drug targets we can identify and the more things we can do to help to manage or treat diseases, right? Okay? All right. Let's talk about our first class of drugs, the statins. All right. These are, as Dr. Naeem will tell you, kind of your gold standard for cholesterol, high cholesterol or hyperlipidemia treatment. Again, they're older drugs, right? But I remember a time, like I said, high cholesterol, hyperlipidemia runs in my family. I remember a time probably in the late 80s, early 90s when my dad was diagnosed with hyperlipidemia. They didn't have statins at that point. He was taking niacin and some other things for it. I remember that very clearly. So again, in the grand scheme of things, these are the oldest drugs in the category, but they haven't been around for 40, 50, 60, 70 years. Okay, but they're very, very effective, and they're the ones we always go to, with regards to hyperlipidemia. And Dr. Naeem is going to talk all about that. You don't need me to tell you anything about that. But let's take a look at the chemical structure here. I want to walk you through two chemical classification schemes, two chemical classes, okay, of statins. We have type 1 statins and type 2 statins. They're classified as type 1 or type 2 based on their chemical structure, not their mechanism of action, not the therapeutic usefulness, not their efficacy, just based upon their structural features. Okay? So let's start with type 1 statins. Okay? There's two examples up here. One which we don't really use clinically, compactin. It was kind of an early type 1 statin, but we do have simvastatin up here, which everybody recognizes, right? I'm sure if you dispense a statin in the pharmacy, you dispense simvastatin. This is a type 1. So let me see if I can use the laser pointer here, or let me see, maybe I can use this. Let me see here. No? All right. So let me use the laser pointer here. So we take a look at type 1 statins. There's two chemical features that are what's the word I'm looking for? That are characteristic of type 1 statins. You can identify very easily. The first one is this decalin ring structure. Decalin meaning 10, okay? Decalin ring structure here, okay? Right here. You can see it here and here in compactin, all right? So that's one structural feature that you see in the type 1 statins, okay? The other feature here that you see, okay, is a part of the molecule here in red, okay? It's a closed lactone ring, and it looks like the substrate for HMG-CoA reductase, which is the target. Down here, you can see right down here, okay? Here's HMG-CoA. That's a substrate for HMG-CoA reductase, the enzyme we're trying to block. And if you take a look up here, almost the same structure as this. This is an open ring. This is a closed ring, okay? So you have the decalin ring structure, and then on the top there, you have that closed lactone ring that mimics HMG-CoA, okay? Now, it's not enough to completely fool the enzyme in that form, okay? What has to happen is an enzyme has to act on this lactone ring to hydrolyze it and open it up. So now it looks like HMG-CoA. So your type 1 statins are essentially, they are prodrugs, okay? They have to be acted on by an enzyme in order for that lactone ring to be hydrolyzed and open up. Everybody got that? Okay?
Which enzyme that is?
Oh, you know what? Boy, I knew somebody was going to ask me that. It's not. Let me look that up. I'm having a senior moment. I can't remember exactly what. I'm having this a lot lately, unfortunately. I will test you on it, but let me check it out. Or if somebody wants to look it up really quick, you can. Doesn't matter. Ask ChatGPT. It's smarter than I am anyway. Okay? But at any rate, so good question. There is again, it is hydrolyzed into its active form, which is just this open ring structure. Okay. Now, and if you find it, let me know what it is. I'm just curious. I can see people typing furiously here. See if you can find it. Now I'm curious. I can't move forward without knowing. Any idea?
Carboxylases, CSE1, and paranoxylases, PON1.
Let's ask ChatGPT. Hey ChatGPT, how you doing?
Hey there, I'm doing great. Thanks for asking. Whatever you got on your mind today.
Yeah, I'm having an embarrassing moment here. I'm in lecture. I'm teaching my students about HMG-CoA reductase inhibitors, especially the type 1s like simvastatin. And I know that drug needs to be enzymatically acted upon in order for it to be activated, and I can't remember the name of the enzyme. Can you help me out with that?
Absolutely. I've got you covered. So the enzyme you're looking for is called a carboxyl esterase. Basically, simvastatin is a prodrug, and it needs to be hydrolyzed by a carboxyl esterase enzyme in the liver to convert it into its active form, which is what actually does the HMG.
Okay, all right, no more showing off here, okay? I'm the teacher, all right? Thank you for your help.
Of course. No show at all intended. You're definitely the expert in the room.
Thank you.
Let me know if there's anything else.
Love ChatGPT, right? It just grovels at you, right? Makes me feel good, right? Carboxyl esterases, all right? Don't you dare tell Dr. Shimoda about that, okay? I'm going to end up getting fired, and you're going to end up talking to this blue circle on the screen all day long, which, again, could be fun. You know, it'll make you feel good. Okay, everybody got that? All right, you like that's good. Okay, now let's take a look at type 2 HMG-CoA reductase inhibitors. You can take a look at these. Just look at them for a second. How are they different from the type 1s? What can you see? What don't they have?
They don't have to.
The Decalin ring structure, right? They've got, again, their core structure there, the ring structures are much more complex, right? All right. What else is different?
They're open.
Exactly. So the HMG-CoA moiety up here, okay, instead of being a closed lactone like it is here, it's already open. Are these statins prodrugs? No. They are ready to go as soon as they're administered. Okay? Now, that's super exciting for me, all right, as a pharmacologist. But from a therapeutic standpoint, does it make a big difference? No. Doesn't matter whether it's a prodrug or whether it's the drug is ready to go. Okay? But from a chemical structural standpoint, you know, it's something to know. All right? Now, some examples of these are fluvastatin, okay, cerivastatin, atorvastatin, rosuvastatin, okay? These are all statins that you learned about. There's a huge list of them in the drug index at the beginning here, okay? I'm not going to ask you, so what I could ask you here from a chemistry standpoint, okay? And did I do you wrong on the chemistry stuff the other day? Were you prepared for what I was going to ask you? I hope you were, okay? I could give you a picture of a statin and say, can you tell me whether it's type 1 or type 2 statin? Okay? Could you identify the part of the statin that is similar to HMG-CoA? Sure you could. Okay? So again, just keep that in mind. That's really what I'm looking for here with this. All right? Let's see here.
Dr. Goy?
Yes.
And maybe you'll tell me something that is like, oh yeah, that's pretty obvious, but right now I don't have it in my mind. Is having a prodrug versus an actively ready drug most of the time not clinically relevant?
That's a great question. can I paraphrase that for you and repeat the question? I think it's a great question. The question was in general, right, about prodrugs, right? Is there any advantage or disadvantage of having a drug that's a prodrug over versus a drug that's been administered and is active in its parent form? Is that what you're asking? You know, I would say, and again, don't take this with a grain of salt, but I would say in most cases, I don't think it really matters unless there's some sort of pharmacogenomic interaction. Okay? So let's say, for example, let's just say pretend, and this is not a real thing, but let's say that I'm a patient and I have a deficiency or a genetic mutation in the carboxylesterases that converts simvastatin to its active form. That drug's not going to work for me, right? So I would have to take these drugs over here, okay? And that's just an example in general, right? You know, with prodrugs, you know, it's a little bit more complicated because usually there's an enzyme or enzymes involved in their activation. And again, there could be a pharmacogenomic issue there where someone has a deficit in that enzyme or a mutation in it, or too much of it or too little of it, right? And that can mess things up. Okay, does that make sense? That's a great question. I think in this particular circumstance, it doesn't really matter. Okay, but it is something always to consider, right? Is, you know, what is unique genetically about your patient, and you'll learn about a lot of the drugs, especially here. Like we talk about warfarin, all right? There's a huge drug right there that's got a lot of pharmacogenomic interactions that you got to watch out for. Okay. Now, let's take a look at, again, this is just a picture of the pharmacophoric structure, of the type 1 statins right here, okay? Just so you can see it. I should have showed it to you before. I kind of forgot this picture was here, okay? Here it shows you the Decalin ring structure, which is referred to as the hydrophobic moiety, okay, of the drug molecule. These ring structures here, whether the Decalin ring structure here in a type 1 statin, or the more complex hydrophobic structures over here in the type 2 statins, we believe that there are van der Waals interactions that occur between those ring structures and aromatic rings in the enzyme, the HMG-CoA reductase, and that's a big part of the binding process that occurs, okay? Is, you know, van der Waals interactions between these rings here in the Decalin ring structure or any of these aromatic rings here, okay? And again, this is just a verbal description of the type 2 statins I talked about, okay? Again, this is just an example. Okay, I don't even remember which one this is. Let me go back here. I think it's atorvastatin, yeah. So here's a picture of atorvastatin, and again, these are some of the types of chemical bonds that form between a drug like atorvastatin and the enzyme HMG-CoA reductase. Now you can just see here, right? There's some van der Waals interactions. There is a dipole bond here that occurs between this fluorine. In fact, this fluorine is really, really important. If we go back and we look at a lot of these type 2 statins, look what they have here. Fluorine, 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 right? Because it's an important binding, important functional group that binds to, okay, a component of the HMG-CoA reductase enzyme, okay? So that's why that's important there, all right? You don't need to know this. I'm not going to ask you questions on this. This is just FYI, so you understand that we have a really good understanding of how these drugs interact chemically with their biological target, which is the HMG-CoA reductase enzyme, okay? All right, this, okay, is really important, okay? Now I'm going to explain to you why in a second here, right? Now, I told you that the statins chemically are classified chemically into type 1 and type 2 statins, right? Based upon their unique chemical structure. But there's also another way to classify our statins, which is way more important for you as clinicians. Don't ever go into a clinical environment and talk about type 1 and type 2 statins. They'll look at you like you've got 13 heads, okay? All right? But you will hear people talk about low intensity, moderate intensity, and high intensity statins, okay? This has to do this is a classification scheme based upon the drug's efficacy. How powerful it is, okay? How powerful it is, okay? Everybody got that? Now, let's take a look at this so you understand it, okay? Statins that are classified as low intensity, okay, mean that the best that they typically can do, okay, is give you about a 30% or less reduction in LDL levels, okay? 30% is not bad, but for someone who's got a real high, LDL levels, that may not be enough, right? Okay? Moderate intensity statins are those that will reduce LDL levels by 30 to 50%, okay? And then high intensity statins, again, anything greater than 50%. So there are these three categories for statins based upon their efficacy or how powerful they are. Okay? And just because, again, a statin is super powerful doesn't mean that you would use it all the time, right? And Dr. Naeem's going to talk all about that. All right, let's take a look at some of these here, just so I can show you. Atorvastatin, okay, and this is also dose dependent. We know, remember basic pharmacology, right? The more drug you give, the greater the biological response, right? There's an upper limit for all of our drugs. So this is another example of dose-response relationships. So atorvastatin at a dose of 10 to 20 milligrams is considered to be a moderate intensity statin because you'll get a 30 to 50% reduction in your LDLs, okay? That's what you can expect from that. Now, is that the highest dose of atorvastatin that you can give? No, you can go up to 40 to 80 milligrams. And at that dose range, what you're looking at as atorvastatin is a high-intensity statin, right? Okay, it has those high-intensity effects. So you can get 50% or greater reduction. So atorvastatin, pretty powerful statin, right? Okay? And again, depending on what dose you get will determine the amount of LDL reduction that you get. All right, everybody got that? Okay? Let's take a look at fluvastatin. Okay? So again, at 20 to 40 milligrams, okay, you get less than 30% reduction in LDL. It's a low-intensity statin at that dose. But if you go up to this upper dose here, if you bump the dose up to the next highest dose, you can get moderate intensity reduction of your LDLs. Okay? Dose dependent. Now, you'll notice that with fluvastatin, that's about as high as you can go. You can't go any higher because you start to get serious, you can get toxicities. Okay? So that's as high as we can go with that. So really, it kind of maxes out as a moderate intensity statin. Does that make sense to everybody? Okay? Simvastatin. And it doesn't matter whether it's a type 1 or type 2 either, right? Simvastatin is what type chemical classification? It is a type 1 or type 2? Type 1. It's type 1, simvastatin. Remember we talked about that? If you look in that couple slides earlier. All right? The rest of these are type 2 statins. But again, 10 milligrams, and you've got that low intensity effect. You can bump it up to 20 to 40 milligrams, and you get a moderate intensity effect. Okay? Dr. Naeem is going to talk to you about this, I'm sure, in her recorded lecture. Okay? My goal here, do you need to memorize these doses? I would never ask you to do that. She might. Okay? Or even, I don't even at this point want you to learn which one of these are high intensity, medium intensity, and low intensity. Okay? All I want you to get from this slide here is the fact that we can classify our statins based upon their efficacy or how powerful they are. And there are three categories: low, moderate, and high-intensity statins. Okay? And they're classified there based upon how much LDL reduction we get. Everybody got that? When Dr. Naeem talks about this, and I'm pretty sure she will, whatever she tells you you need to know, and her doses might be different than mine, go with hers. Okay? They should be the same. But if there is any discrepancies or differences, go with hers. I'm not worried about that. I just put that up there so you could see how if we bump the dose of a statin up, we can go from low intensity to moderate intensity or moderate to high intensity. Okay? Because it's a dose-dependent effect. Everybody got that? Any questions about that? Yes, sir.
Can you shed light on the reason why atorvastatin, simvastatin, and rosuvastatin are in our top 200? I assume in part because we have the two high intensity ones.
Yeah.
But other than that,
Boy, that's a good question, right? Because, and you'll see this a lot with drug classes that have got a lot of different examples in it. So, Jose, I'll tell you right off the bat, I don't know, okay? But I can guess, there's usually a lot of different reasons, right? It could be cost, it could be availability, it could be marketing, right? Like, for example, simvastatin's been out a long time, right? And it's a really, that's what I started on. Back when I was in my early 30s, I was started on simvastatin because it's a well-known kind of low-intensity type of statin. But when I realized that it wasn't being controlled, okay, I got bumped up to atorvastatin, okay? And recently, I'm up in the 40 milligram range. So again, as I progressed, I went on different statins and different doses depending on how well controlled my LDLs were, okay? But again, it could be a lot of different things. Like I said, availability, marketing, cost, just what was up first, right? Like I think atorvastatin was one of the first high intensity statins out. So again, the first release is usually, or maybe it went generic first, right? Or whatever. So there's a lot of different reasons for that. I don't know. I could ask ChatGPT again, but I don't want to be embarrassed. So, but that's it's a good question.
That was interesting because
it is very.
A bunch of things that might be behind.
It could be a lot of things. Absolutely. Any other questions? These are great questions. I appreciate that. And that actually might be a really great question to ask Dr. Naimi too, if you see her. She may know. Okay? All right. Any questions about the intensity piece? Okay. Perfect. How do these drugs work? And Jess, I got 10 minutes left. Okay? You let me know. All right? All right. This looks familiar because we just talked about this in the last hour, right? So again, all this is showing is the cholesterol biosynthesis pathway. Our statin drugs inhibit the second enzyme in that pathway, HMG-CoA reductase. What that does is it does a couple of things. Directly, it shuts down cholesterol biosynthesis in the cell. All right, and the cell still needs cholesterol in order to function. So if it can't make it, what's it going to do? It's going to look for it in the blood. All right? Right? And so in response to that, low cholesterol production inside the cell is going to produce, okay, more LDL receptors, okay, and start to capture more LDL particles out of the bloodstream. And that's where we get the lowering of plasma LDLs from, okay? The inhibition of cholesterol biosynthesis in the cell is kind of a direct effect of the drug, but it is the consequence of that ultimately leads to reduction of plasma LDL levels, okay? Because the cells are now going to pull LDL particles out of the blood because they need cholesterol because they're not making it anymore, okay? Does that make sense? All right. Yes, sir.
That inhibition of CDH
I'm sorry?
the inhibition of the enzyme.
Yeah.
Is it an irreversible inhibition?
I think it's a reversible inhibition. Yeah, I don't think it's
The
irreversible.
liver doesn't react by alternative pathway or something.
In terms of cholesterol synthesis, not that, so again, this is what's pictured here is actually a liver cell. Okay? So again, the liver is important in a lot of ways for capturing LDL particles, but so this happens in adipose tissue, skeletal muscle tissue as well as process. Again, these statins can cause liver damage, okay? And I think it's more due to the way that the drug is metabolized and eliminated from the body versus anything like this, okay? Does that make sense, Kingsley? All right? That's a good question. But in terms of the liver kind of responding to that, I think that again the big response is it's going to be pulling more LDL particles out of the blood and okay. All right. Side effects. There are several side effects associated with the statins, but the big one we're going to talk about is myopathies. Okay, muscle. You guys have all heard about muscle pain, right? The issue with them, right? There's a couple of different types here. We've got myositis, okay, which is inflammation of the skeletal muscle. All right, it's not, so and it can cause pain, muscle pain, in conjunction with statin usage. This by itself isn't a real problem. Dr. Naeem is going to tell you all about how to manage that in patients and what to do if they start experiencing skeletal muscle pain, all right, due to this. And there's things that you have to do and watch out for. But what I wanted to point out with is rhabdomyolysis. This is very serious. This is kind of an extension of myositis. It's when this really goes bad, this whole process. And what happens is that muscle tissue begins to break down, okay? And this releases myoglobin into the bloodstream. And what that can do is it can clog up the nephrons and really wreak havoc and damage your kidneys. So rhabdomyolysis is something that's very, very serious and requires immediate medical attention and IV hydration, I believe, in order to protect the kidneys and prevent the glomeruli from getting clogged up with this myoglobin protein, okay? Because once that happens, the nephrons are dead, okay? So you gotta watch out for that. Yes,
I'm just
George.
curious about if you how the drug box should have inside of that.
Yeah, we're not really sure, to tell you the truth. So that's a really good question. Why does this happen? It's not really well understood, but it does. And again, Dr. Naeem will tell you about what percentage of patients this can happen in, but it's something to definitely watch out for. That's why, again, there's steps you have to take that I want her to explain to you, right, about what to do if a patient experiences kind of muscle pain and inflammation, because that could be a sign that this patient could be predisposed to rhabdomyolysis. It could progress into rhabdomyolysis, which is not good. Yes.
So I was thinking, is this a matter of like the kidney not being able to keep up? Like if this happened like in a lower pace, would the kidneys be able to?
Yeah, you know what? I think it has to do with certainly how quickly it happens and the volume of protein that's released into the bloodstream that the kidneys have to deal with. Okay, again, we know that high levels of plasma proteins, right? You have high protein diet or things like that, you take protein supplements, that over time can damage your kidneys. I think this is such a massive release of myoglobin that the kidneys just can't handle it. They get clogged up, and it's a mess. And but again, there's ways to deal with that and to limit or prevent the kidney damage, especially if you catch this happen. I think your urine actually turns like a brown color with this. If I remember, don't quote me on that. Dr. Naeem will probably talk about it. Okay, but yeah, definitely myopathy. And again, not really sure why this happens, but it does with the statins, okay? All right, study stop what we got here, okay? All right. 1, 2, 3, 4. About 8 slides. I'm counting how many slides we have left, right? Right? All right, so here's what we're going to do, right? And what time is class supposed to be tomorrow? Did we get that straightened out?
1:30.
Is it 1:30? Okay. Can you give me 1 second before I let you go?
Yes.
We did pretty good today. All right. I could have plowed through this, but we're not going to do that. 1:30. Okay. I will send you around a Zoom link. Okay, and what we'll do, if you don't want to attend, you don't have to. I would love to see you there. It will take me probably about 15 or 20 minutes to get through this. Okay, and if I remember, I'll try to record it to make sure that we've got it recorded. I don't think you have to listen to too many recordings, but can you? I'll send it out, and hopefully I'll see some of your smiling faces, maybe all of them. Okay, tomorrow afternoon from the comfort of, and you don't have to wear professional. You have to wear clothes, but you have to, right? You don't have to wear professional clothing. Okay, be comfortable. All right? We'll see you guys tomorrow. Yes. What's that? Yeah, hold on one second.